Hello everyone. So the next topic which we will be discussing today is about the cell cycle. So in this cell cycle is basically an ordered series of events. So it is a sequence of events by which a cell duplicates its genome and even actually divides into the two daughter cells. So in this we will be discussing the main two phases of cell cycle, the diagrammatic representation of the cell cycle, and what are the functions, the specific functions. For example, there is M phase, there is G2 phase, there is S phase. So what are the specific functions of this? So let's get started with the first slide of cell cycle. So it is an ordered series of events, and it is a sequence by which a cell duplicates its genome and will even actually divides into the two daughter cells. So there are two main phases which we will be discussing. It is the interphase and the M phase. So interphase is the time during which the cell is preparing for the division by undergoing both the cell growth and the DNA replication in an orderly way. So the interphase is subdivided into G1 phase, S phase and G2 phase. So in this, G1 phase is the gap 1 phase. So G1 phase is also called as gap 1 phase. In this, it is a period which is between the end of the M phase and the start of the DNA replication. So it is the gap 1 phase. Then the next phase is synthesis phase. S phase, which means synthesis phase. In this, the period during which the DNA synthesis occurs. So this is a period during which the start of the DNA synthesis occurs. Then comes the G2 phase. In G2 phase, which is also called as gap 2 phase, it is the period which is following the DNA replication and preceding the initiation of the M phase. So interphase includes, interphase is basically subdivided into these three phases. And after this comes the M phase. We were discussing that in this the period it is between the end of M phase and the start of DNA replication. So M phase is here. So M phase, period of actual division, which is corresponding to the visible mitosis, mitosis phase. So this is the M phase. We will be discussing in detail about all these phases. So the cells, they do not enter into G0 phase. So there is a G0 phase also. So what is happening is the cells, they do not enter into the G0 phase and they enter into G0 states and that is being divided into two parts. That is reversible resting state and irreversible state. So we will be discussing in detail that what are these G0 states which are subdivided into two parts. So this is the reversible resting phase state. This or quiescent phase. So the cells that remain metabolically active but there is no longer proliferation and it will depend on the requirement of the organism. So this particular state, it may take up to days, it may take up to years before resuming the proliferation. So cells in this particular phase, this is also called as reversible resting phase. The cells are metabolically active but they no longer proliferate. The proliferation of the cells is not happening but they are metabolically active. So that's why this phase can remain for days, weeks or even years before resuming the proliferation. Then about the irreversible phase. We are discussing here the two things in this also, the senescent and terminally differentiated. So what is happening is senescent cells, they are dysfunctional and that has ceased the proliferation and permanently withdrawn from the cell cycle. So senescence means the death. So here in this irreversible phase, in the G0 states, they are dysfunctional and they are permanently withdrawn from the cell cycle. The next, in the irreversible only, there is a terminally differentiated cells. So cells in the course of acquiring specialized function have irreversibly lost its ability to proliferate. So in this, they have lost their ability to proliferate. In the reversible, they do not proliferate, but they were metabolically active. But in the irreversible terminally differentiated cells, uh, they lost their ability to proliferate. And the example of this is the mammalian skeletal muscle and the nerve cell. So they will not proliferate they will not proliferate further. Next is the diagrammatic representation. Here what we are showing is there are four phases which are being depicted here in the case of cell cycle. So what is happening? There is a G0 phase first, then comes the G1 phase. G1 phase is the growth phase. Then comes the S phase where the DNA synthesis takes place. We have also discussed in the interface zone only that interface zone consists of G1 phase. So if we are talking about interface, then we are talking about three that are being subdivided into these. Interface is being subdivided into three categories. That is G1 phase, S phase, and G2 phase. So G1, S, and G2, this is the part of interface. So this is the part of interface. So here S is the period during which the DNA synthesis occurs. G1, it is the period at the end of the M phase and start of the DNA replication. So M phase is ending here, M phase is ending here, and there is the start of DNA synthesis or DNA replication which has been occurring. So it is the phase, but this is the phase. G1 phase is this phase. Then comes the G2 phase where it is period between the DNA replication and proceeding to the initiation of the M phase. So here the DNA replication is ending and it is the initiation of the M phase. So this is the G2 phase. So G2 phase is responsible for the growth and preparation of the mitosis, and M phase is the mitosis, which means cell division. So this was about the diagrammatic representation of the cell cycle. Then what next comes is we have discussed all the phases. We have discussed that in G0 phase there are two types of reversible and irreversible states, and most of the cells in our body are in this G0 state only. So most of the cells in our body are in the G0 state only. Next is approximately 95% of the cell cycle is spent in the interphase zone. Interphase, which has been subdivided into G1, S, and G2 phase. So the duration of three stages varies from species to species and from cell to cell within a species. In most, the whole M phase takes an hour, which is a small fraction of the total cell time. So M phase approximately takes an hour. Then these are some of the time durations that how a cell cycle is being taken place in the body. So rapidly proliferating human cell culture with total cell cycle time of approximately 24 hours. The G1 phase might last for 11 hours. S phase might S phase about, which means DNA synthesis, lasts for about 8 hours. Then G2 phase for about 4 hours, and M phase will be about the 1 hour. So these are some of the time durations during which the whole process of this is being taken place. Then interphase is also called as the resting phase, but it is considered as the most active phase during which the cell is preparing for division by undergoing both the cell growth and the DNA replication. So it is the most active phase. S phase S phase I have already told, this is the synthesis phase, so in which the DNA synthesis or DNA duplication takes place. Then M phase, it starts with the nuclear division. So here we are introducing two terms, that is karyokinesis and cytokinesis. So Cairo has been derived from the nucleus only, so it is the formation of the nuclei, daughter nuclei. That is why it is called the karyokinesis. And cytokinesis, it is basically the division of cytoplasm, because the word cyto is being depicted here and kinesis means to break down. So division of the cytoplasm, and karyokinesis is the formation of the daughter nuclei. So M phase starts with the nuclear division, which is corresponding to the formation of the daughter nuclei, which is called as karyokinesis, and they usually ends with the division of the cytoplasm, which is also called as cytokinesis. So this was all about the cell cycle. I have described all the phases of the cell cycle, their function, their time duration, and this is the reference from where I have taken the content and made the slides.